So then I see we are a minute after the hour, so we'll go ahead and get started. Good afternoon, everyone. My name is Ashley Ferreira, Director of Education, and on behalf of the FSHD Society, welcome to today's FSHD University. Before we get started, just a quick reminder, this session will be recorded and made available on our YouTube channel within the next week or so, so please make sure that you are on the lookout for that. We also encourage you to use the chat feature so that you can let us know where you're from, interact with each other, please. But we do want you to know that if you have any questions, please use the Q&A section just because that is where we're going to be monitoring them and we want to make sure we don't miss any questions. So, in today's FSHD University webinar, Dr. Seth Friedman, Manager of Innovation, Imaging, and Simulation Modeling and API at the Center for Respiratory Biology and Therapeutics at Seattle Children's Hospital, will discuss the role of MRI in FSHD clinical trials and how advanced imaging helps track disease progression, select trial participants, and evaluate therapeutic impact. I'm also very excited that today Dr. Lucienne Ronco will be joining us to help guide and moderate our Q&A session. So again, if you have any questions throughout, please make sure that you use that Q&A function. And without further ado, we will hand it over to Dr. Friedman.
Thanks so much. Can everyone see my slides okay?
Yes.
Okay, super. I was like, what could I possibly be sharing? But so thank you so much. So today I'm really going to try and cover sort of everything MRI biomarkers and just walk you through the last many, many years of research. Let's see if I have control here. Okay, here are my disclosures. And I really want to kind of preface my talk with a science story that I absolutely love. This of Arthur Worthington. Maybe some of you have heard of him. So he was a scientist that studied splashes, like when you drop a splash on a horizontal plate. And in the late 1800s, he was doing this by trying to visually observe them and draw the splashes. And so at the left here, you see a publication in 1877 on the forms assumed by drops of liquids falling on a horizontal plate. And so at that point, he believed that everything that he was seeing was symmetrical. And what was remarkable about Arthur Worthington, the time he lived, is that photography came about in the next 15 or so years. And in 1895, he published another set of papers in which he discovered that all the splashes were in fact asymmetrical. And this is something I really feel like you're going to kind of get a sense of as we go along the different avenues of MRI research and MRI and function, is that kind of everything seems very homogeneous. And then the more we look and the more tools that we have, we start to see that everything is more kind of like these splashes on the right-hand side. So just to briefly review, so there are obviously many, many different kinds of images you can get out of MRI and many different types of contrasts of water. The main ones that we use are Dixon imaging, which can derive two main sequences, one where you get an image of water and one that you get an image of fat. And you can see on the bottom here, this is a slice through the calf, that there is fat near the bone at the top in the tibialis anterior, and in the medial part, which is the medial gastroc. And it's much more on one side than another. The other sequence that we use a lot is short tau inversion recovery imaging, or STIR. And STIR really has a pretty cool capacity to null all the fat signal. And then what you're really seeing is free water. And in this patient, you can see there's quite a bright blush in the center of the medial gastroc. And this is what we've oftentimes termed STIR plus or STIR positive. So these are the two kind of main sequences we'll talk about today. There have been a lot of other things used, T2 relaxometry to measure water. We've used ultra short TE sequences to look at things like fibrosis, but these are sort of a little more niche and not as common across the field. And so to kind of parallel like where we were 15 or 20 years ago, it was quite difficult to measure a lot of things on muscles because it was a very manual process. And this is from Laura Ream's paper in Scientific Reports, where now we're really able to segment the muscles across the full length. So at left here, you can see a shrink-wrapped blue shape on top of the vastus. And from this, we can derive all sorts of volume metrics like muscle volume, fat fraction. You can then also convert this to more of a linear plot where you're looking from one side of the muscle to the other. And so in the second plot B, fat is yellow, muscle is red, and fat fraction is black. So you can kind of follow these things along the muscle. And then we also have the ability now to make these really exquisite pixel maps where we can map fat back onto the muscle, just the raw muscle data. And from these, we can start to make what we call fat pattern metrics. And these will become increasingly important, as you'll see as I go through my talk. They really haven't been studied that much, but you can imagine deriving a standard deviation, like a fat variation measure. You could look at the distribution of pixels and get an idea if it's skewed to one side or the other, which would happen with fat. It could become more blobby, sort of more flattened, and this is a kurtosis measure. And you can also derive measures of spatial autocorrelation, something called Moran's eye. So we're going to kind of look at these and kind of what their role is in the disease. So this is what I hope to cover today. I'm going to kind of give you a good overview of like what we know about fat and stir in the body. Then we're going to talk about fat patterns within a muscle, these kind of pixel maps. We're going to really hopefully clarify what predicts progression. So if you have a patient at baseline and you're trying to see where they're going to be in 1 year, what the strategies are for that. We're going to focus a little bit on whether fat location matters and how that could affect something like strength. And then I'm going to delve a little bit into these more complex function tasks that probably many of you on the call have done, and certainly we have in our batteries, things like walking, picking up a penny, time up from a chair, or reachable workspace. And we'll hopefully have time at the end for summary and for questions. So my journey, just very briefly with FSHD, began almost 20 years ago. And Dennis Shaw, I just thought I'd embarrass him here. I know he's not on the call. He's a radiologist at Seattle Children's, and he invited me to coffee, which is always a tell for something's gonna, you're gonna get asked to do something. And at this time, he said, "Hey, how'd you like to write a grant in FSHD?" And at that point, I knew nothing about what FSHD was. I mainly studied muscle biochemistry at the time. I'm sorry, brain biochemistry, all above the nose. And I remember looking at this picture on the MDA website and thinking, okay, it affects these muscles. Well, let's do a study in the legs because there's just some down in the, you know, it's not even the TA in this picture, it's something else, but we'll study that. And so we applied to Friends of FSH and we got a small grant to begin doing some imaging and trying to quantify what biomarkers were present in the legs. And I think what I was most struck with, just not knowing as much about the disease, is how asymmetrical things were. And so at the top here on the left, you can see the right and the left leg of an individual, where the left leg has a lot of fat deposition inside the muscle. You can see the white areas. And the right leg looks pretty normal by all intensive purposes on the T1 image. But both images have some of this bright stir signal on the bottom panel. And you can notice that on the left leg, it's almost all fatty replaced where the arrow is. And so some of this was also a question at the time of like, how much of stir signal is in muscle? How much is in fat? And can we kind of detrend those things? And at this time, we used a very kind of 32 echo T2 series to try and create some automated method, which was exciting, but it didn't cover that much of the body. And as studies started to amass where there really were studying large cohorts across large regions of the body, this is a study out of Italy, that Giorgio Tosca's work. And you can see here, as you look from the core down to the legs, there's kind of this topography, almost this mountain range of which muscles are affected and which muscles are less affected. And this was really notable because I think that this is what visually we all saw, but it was reinforcing to see it in a plot where you really have some muscles like the popliteus and the tibialis posterior that are quite low incidence. And you have other muscles. I'm not sure if my mouse can be seen, but like one of the highest plots here is the semimembranosus, which is in the hamstring. So lots of variability. What they also rated is they tried to evaluate how many of these muscles had bright stir signal. And this was the two arrows that you see on the right, where when muscles had fat, about 6% of the time, they also had some stir signal. Not necessarily clear whether it's coming from muscle or fat, but that was sort of the phenotype that they saw in this study. Here's another study out of the Netherlands, Carleen Moel, where this was a large cohort of both FSHD1 and some FSHD2 patients. You can see them denoted by the little 2 in the panel, in the column. And the fat labeled column, you can really see that there's quite a similar kind of light bright, many muscles in the lower legs that are affected in this cohort. And I just want to call out that a lot of these studies have measured left and right, and you could imagine if left and right were equivalently affected, you might see a very similar pattern between the colors, but there's a lot of speckling in this heat map, really showing off that there's a lot of left-right asymmetry in these patients. And they saw a very similar amount of stir elevation on the right side of the panel, about 3.5% across all the muscles that are being affected. So we're starting to get this understanding that fat's very pervasive, stirs in some muscles. And as you look down this plot, something like the TA, which is sort of halfway through the stir column, it does seem that there's some muscles that are more vulnerable. And it's still not clear whether this is if the muscle that points your toes is affected and you have more trouble walking, that muscle's just getting loaded all the time and is stirred right, or if really there's something pathological going on in some of those muscles where it's a disease sort of driver or a disease result. I think probably some of both are true. And then as studies started to move into whole body, this is Doris Lung's study out of Johns Hopkins, where they rank their patients sort of from least affected to most affected on the right. And you can see a very similar as definitely as you become more and more affected towards lack of mobility, you have a lot of red, so a lot of fat fraction. But nonetheless, I think it's clear that patterns here is heterogeneity versus having homogeneity. In our own work, this is data from the data lake that was initially funded by friends, and now we have a lot of contributors. Um, and so what's notable about this, so this is all the muscles that you can measure basically in the body at present. And Springbok has a database of normal subjects, and you can see kind of the gray mountain range at the very bottom of the plot, right around 10%, which is normal fat. So this is the normal fat fraction that you would have in a population. And the dots are really the mean fat across the entire cohort, which is in the data lake. And these are also ranked by sort of how much fat is typically in the muscles. And at right, it's difficult to read, but one of the hamstring muscles, the semimembranosus, tends to be very commonly affected across a cohort. And at the left-hand side, something like the popliteus or the tibialis posterior are much less affected. And you can see within this sort of spread of the bars, there's quite a broad range. And so within a population, almost all muscles are affected, but it's not necessarily true that it will always be the semimembranosus and never be the tibialis posterior. And I always, whenever I give this sort of a slide, I hear Doris's voice in my ear, because every time I see an image that I'm like, oh, it's so amazing. We finally have the tibialis posterior affected without the, fill in the blank. Doris has seen so many clinical images that she's always like, oh yeah, I've seen that before. That's not that uncommon. So we tend to think that these things are very uncommon, but it's just sort of pattern variability is the norm. And we recently took some of the MOVE Plus subjects. This is the big study out of the CTRN that probably if they're investigators in this call, many of you may be involved in. This just shows that if you take a new sample and you overlay this onto the data lake, you get a very similar spread. And I just want to call out that if you compare the upper and the lower body, we for many years at our center just studied the lower body because that was kind of the imaging bias that we had. And it's very clear from this that muscles both in the upper body and the lower body are kind of distributed across this graph. And so this really is a whole body disease that I think now, based on a lot of the early trial work that Fulcrum started and others, like looking at the whole body is the right strategy to try to follow what's going on. And so, this basic pattern of heterogeneity ruling between muscles and across individuals is very true, but we really wanted to start understanding what's happening within muscles. And this was really something that was, an epiphenomenon of the analytic method that Springbok has developed, which is now we get all these beautiful pixel maps to try and say, what is going on? And I should mention there's another study, Linda Heskamp out of the Netherlands published a paper in 2022 that has similar plots that are really, really interesting. And so to go back to Worthington for a moment, one of the things that you see in the literature and we have contributed to, and it is repeated over and over and over again, is that the muscles fill up from the bottom to the top like a cup, this distal to proximal gradient. And I just highlight our paper here. This is a figure from our paper where you can see at the bottom arrow in the center of the screen, there's a lot of fat in the medial gastroc. And as you walk up the muscle towards the knee, you can see that there's much less fat. And so this has been something that is sort of concluded over and over again that this is the way it is. All muscles behave this way. And while it is certainly true that some muscles proceed in this type of pattern, what we've been trying to characterize is sort of all the patterns that are present. And this is Laura Ream's paper that was published in Scientific Reports. And I don't know, this is still something that I'm so excited just to be seeing these kind of changes, because I think we always looked at the images and we tried to infer visually what was going on, and it was quite difficult. So here's some examples. So within the biceps femoris, here's a subject that starts with a fairly uninvolved muscle, and over a year, it progresses quite rapidly across the entire muscle. The next is the soleus in the calf, where you see it. This is sort of a distal to proximal kind of a change. The sartorius goes from the top down. The tibialis anterior in this example goes from the outside in, so the top and the bottom are affected, and it kind of creeps towards the center. And then you also can have from the center out. Within some muscles, we can see that one muscle is going up, one muscle is going down. Here's an example of the gracilis. And then within the tibialis anterior, we can have situations where this is plotting all muscles in a cohort in our Wellstone sample, where you can see that you kind of have all patterns within the TA. And I think this is important to just comment on, is that these are not phenotypes. The plot I showed you a couple slides ago, the soleus doesn't always go from the bottom to the top. Many of these patterns are present in an individual muscle, and I think there's still a lot of work to do to understand why. And so this would sort of be my summary of this sort of what we know so far, is that these initial kind of simplified diagrams that's on the MDA website even today of FSHD is clearly very simple. Wikipedia is a little better. At least it gives you the back, so you get to have a hamstring muscle. I've not edited Wikipedia, but this did make me think about it on pulling this figure. And really, when you overlay the muscles, everything is involved. But I think the trick and the challenge of the disease is we're really trying to understand individual progression. And so that's sort of, I think, is really where we've been focused on, is like these commonalities are nice, but really we're trying to predict an individual over time. And so how to best follow progression. So there have really been three main strategies. The old strategy that I think we've all participated in is just like, let's lump the muscles together and see if that will predict change. And then there's sort of been a shift over the last, certainly in the last 10 years of like, let's take a look at muscles that have some phenotype. Um, Fulcrum used this moniker of B, like things that had moderate fat fraction. Many studies, including things that we've published, have really talked about that this moderate group of muscles at baseline seem to be more likely to progress. And then really what we're kind of advocating for today and what we're still working to operationalize is this individual muscle approach. And let me kind of walk through. I tend to draw these little cartoons that I think they're equally confusing, but maybe helpful. But so the all approach at the top is kind of you take a human, you take all the muscles that you've measured, and you put them into a soup pot. And that soup pot maybe says, okay, this bunch of muscles is 20% fatty, and then you look over time and you see that one muscle maybe in that group has changed, but we're not worried about that anymore because we're just looking at the soup, and you have a very small change because most don't do anything. So that's kind of the regression to the mean, maybe worst approach. Type B really was a desire to say, okay, let's take these muscles that are likely to change, let's just put those in the soup pot and let's follow those over time. And that does give you a boost in your sensitivity. The only dilemma is it kind of treats all muscles as the same. And I think what I'm going to show you now is that the muscle identity makes a big difference in terms of how the change occurs. The other thing is you can look at my little drawing in the center there on the left is that there's a lot of red dots that are not included in the pot. And so we're really leaving out a lot of muscles. And there are muscles that are going to be changing within those red dots that we're going to ignore. And so this approach kind of suffers from treating all muscles the same and then not including things that might actually have important variants. And the last that we've sort of been really working with over the last year is this idea of a random forest where you put all the data in. You can also add in other data, such as your D4Z4 length or your age or your gender. And you put all the data in and you're leaving things like muscle identity in the model. And so you're able to actually evaluate if those things are useful, and you're able to kind of solve the question of what's happening over time. So let me just walk you through this real quickly. And um, I think I have Sylvia's citation here in one of these slides. So this is basically this idea that you take all this data and, one of the pieces, number two here, is that we're actually going to classify the muscles. So the TA is different than the vastus, is different than the soleus, is different than and so that is actually a component of the muscle. And we're going to put all these things in at the same time. And the way you do this is you use a training set and a test set. And what we're doing is we're trying to predict change in lean muscle volume or fat fraction over 1 year. So the other thing that Sylvia did, as a biomechanical engineer who looks at muscle, I think that for a lot of times my emphasis was always radiologic, and we've had a lot of neurologic focus as well in the disease, but I haven't really had as much biomechanical focus in how to solve these problems. And one of the things I think that is super interesting about this model that you'll see why it's so valuable is that first the muscles are divided up into pieces. And this allows you to really boost your signal to noise in a small region that's changing over time. And you can imagine in a larger muscle, if there's only a tiny blob of fat, it takes a lot of change to have sensitivity. This also has a bunch of functional model things in it, and we can put tasks in. I won't go into as much detail, but I'll give just some examples. So we're really trying to predict fat change and also like just change in the human over time. And what do you get out? I should have warned you, this is a complicated plot. So it's a little different than how you think about normal correlation, where I think we're all used to seeing a correlation plot, which is a variable on the x-axis, a variable on the y, and a nice linear method. So what we really get out here is we get terms out of the model that are significant. And there's two things I want to highlight. And so in the middle frame here, this is called a SHAP value, S-H-A-P. So when we look at the regional model, the model where all the muscles are cut up into pieces, what we see is that the pattern of fat makes a big difference in prediction. So that's one thing I want to emphasize is that, you know, having a smaller chunk and knowing the pattern is very predictive of how the patient is going to change over time. When you step up to the individual muscle level, this is where we can start to look at other variables like D4Z4, which is underlined in the top right. But the most important thing to come out of this phase of the model is that muscle identity makes a big difference. And so knowing that a muscle is a semimembranosus is very helpful compared to knowing that a muscle is a popliteus. And so the reason this is important is that it sort of suggests that these strategies to lump things together will probably not help your prediction. And you can also generate these kind of plots where on the x-axis is the measured change. So for example, Billy enters a trial and we follow Billy at 1 year and we can plot that data versus the model. So this is the data that has come out from our random forest model. And you can see all of them are pretty well correlated. And so what this is suggesting is that we can put all of these variables into the model: muscle identity, fat fraction, bone volume. And what these allow us to derive is a very good prediction of where the subject would end up if we didn't have their measured data. And so this was really exciting because I think this was always the hope is we can take this complicated data, we can predict change, and we don't have to lump it into a soup pot where we kind of take out a lot of the complexity. And so as we kind of have been going on then, we sort of wanted to test these things about different, just different ideas that we've had. And one of the persistent questions has been, how good is MRI really compared to DUX4 at predicting progression? And so this is a study we've done recently where we took a sample that we have from the Wellstone, where we have DUX4 data from a central biopsy at baseline, and we're able to make these similar random forest models to try and look at, is MRI useful at predicting progression versus DUX4? Is pathology useful? Is CSS useful? Kind of put everything in the mix and see what we get out. And so this is a similar SHAP plot, but it's just displayed in a different way. And so what came out of this model is that the most predictive feature to predict fat progression over 1 year in the tibialis anterior is really the pixel variation. So this fat variation pattern. And you see here, there's kind of like a stepped waterfall progression, and I'll just kind of walk through those very quickly. So the next batch of these really are similar pattern variables. And so it was certainly very interesting to us that the best predictor of change over time is probably the fat pattern that exists in the muscle. Knowing where the muscle starts in terms of fat percent is also helpful. And then it takes a couple kind of stair steps down to get things like immunoglobulin, quantitative stir, DUX4, and then we can walk down all these other variables. And so, one comment I want to make is this is really a model that's built up of all of these terms. This is not a simple model where we say it's just fat variation. And so some of the next phase of this project is really model testing and model building to determine the best sort of most parsimonious group of things for prediction. But it's pretty intriguing that fat variation is so useful, and it's something that I think we have to build into future model making. So I want to move on to function, and I'm going to kind of take a similar approach. So, function has also been this sort of thing within FSHD where we tend to lump everything together and we try and understand what's correlated to what. And so this is some data from Carlene Moule, and I think probably every group that has studied FSHD has made plots identical to this in their papers. We have many. And on the left, you see you have fat fraction in the legs on the y-axis. And on the x-axis, you have a motor function measure, and they look very correlated. And the same is true for the 6-minute walk test. And so this phenomenon has really been used to say, aha, if we can correlate body fat with function, we're good. Like, no problem. This is how we're going to proceed with the disease. And one of the challenges is that it's not always so simple when you start to look at more strength-based measures. And so this is something that we looked at in our Wellstone sample, where we looked at TA strength. And we always had these funny kind of non-linear relationships where it seemed like people were doing pretty well with their strength, and then they started to get more fat, and then things precipitously dropped off the cliff. And at least in talking to Sylvia and her colleagues about this, we had this notion of like, okay, well, maybe it is just the amount of fat that creates this phenomenon. But we'd never really been able to do any research looking at location. And so this question of, well, maybe the pattern has more meaning than simply, oh, it's just this is a top to bottom, bottom to top. And so in Sylvia's lab, they had done lots of work in the past where they had tried to evaluate where fat can be and how that affects different functional performance. And so this was a study where they created model kind of muscles, and you can see the muscle has this kind of very non-biological shape, but it has these different conditions of a sheet inside, it's got a fat block at the end, it's got fat throughout. And so this seemed like a pretty good proxy to try and use for our muscular dystrophy data. And so, continuing to learn things about muscle as a neuroscience person has been definitely my last 15 years. And one of the things that was remarkable to me is that you could actually divide up the tibialis anterior into these different heads and aponeurosis. And so you could segment it to start to look at where fat was in our Wellstone patients and how that was affecting function. And so they use a bunch of finite element modeling. So we took our segmentation, and then they basically perform a lot of what if kind of scenarios. And this is what the data shows. So on the left here, you see 2 patients segmented tibialis anterior muscles. So these are the muscles that point your toes. And you see 2 very different fat patterns. So you see one person has kind of wispy fat towards the bottom, and another patient has fat kind of all along the, one side of the TA. And interestingly, neither of these patterns really affected force at all. And so, we're trying to say, okay, well, what creates this nonlinear relationship? And so what they found is that there really is one pattern that more so than others that will cause this rapid drop off in strength. And I will forever give Doris credit for this, but Doris calls this the mozzarella cheese stick phenomenon, which is, if you take a mozzarella cheese stick and you actually pull it apart fiber by fiber, it still pretty much functions as a mozzarella cheese stick. But if you bite the mozzarella cheese stick in the middle, if you affect the length of the muscle, this is how it really impacts its function, given it's a long, skinny muscle that contracts across its length. And so this is sort of, I think, an interesting method that we hope to do more of to really try and start to understand how does the location of fat relate to function, as opposed to just saying 10% is 10% is 10% is 10%, which clearly in some of these discrete tasks will not be the case. What about more complex functions? So I think this has been on everyone's mind, which is these are the, dorsiflexion strength, great. Probably from a patient perspective, this is meaningless. These more complex functions where task-based effort or task-based performance is really what we're trying to predict, because that has the most relevance to the patient and to potentially following change over time. And similarly, this is again, these are plots that we've all made, and this is Michelle Mellion's work from neurology, as part of the Fulcrum study. And, here's time up and go where you get up from a chair. And on the x-axis, you have the fat fraction. And you see this very nice linear relationship where the more fat that you have, the longer it takes you to get up and out of a chair. So I think we can all agree, like this makes total sense within so sort of the human form. But what you can imagine it's quite difficult to do is if you want to measure change over time in this is probably not the best approach. And indeed, in our data lake, when you rank the tug, so on the bottom axis here, you can see we've taken all the patients in the data lake at this point, and we have put their tug score on the x-axis. And you can see that there are some patients who have a lot of red in these whole body fat maps that are unable to do the tug. And then there's a precipitous drop in some patients. And then for a long, long run, there's just very minimal differences in time from like 8 seconds to 10 seconds. And so, one of the demonstrations of this random forest model is actually what we wanted to understand is could we use it to tell us which muscles were involved and how to predict who is going to change over time. And so this generates another set of SHAP plots. So this was a small subsample that had tug data at baseline and over time. And what we were trying to do is to see is could we use the random forest model and these individual muscle measures to predict whether or not the person would change. And what we found is that there were some muscles that were much more predictive of change over time than others. And these are things like at the bottom, you have the trunk extensors, so your ability to kind of posturally brace. And then a range of hip flexors, the extensors, and the abductors. And then the knee extensors, this ability to kind of generate force. And I've drawn my little cartoon on the right here. And so, for me, like thinking about this and like, okay, what do we do with this data? Well, this is a good example of if a patient has these muscles affected, this would be a useful task to look at in a 1-year time period. If none of these muscles are affected, you're probably not going to see a change in tug over time. And so this ability to predict change in assemblies of muscles is very helpful in how we think about following the disease. What about more complicated tasks? And so this is something where I think reachable workspace has been on everyone's mind in lots of different ways. It's a very interesting computerized task that has components of the tug, but certainly it's more quantifiable. So here's another example where if you look at the total fat fraction again, you see this inverse relationship where you're essentially doing better the more muscle that you have. And in starting to work on this with the whole body MRI that we have, I really feel like there have been this learning of who's doing the test makes a big difference, and what the task components are not always clear. And so let me just show you this and talk you through this plot, because this is, I think, data that Doris just presented in Italy, but it is sort of fascinating and it gives us some examples of how to proceed forward. So on the left here in the green box, you have all the different muscles are represented on the bottom on the x-axis. And so can you see my cursor, Lucienne? I can't tell oh, you can. Okay. So the reachable workspace. So everyone that is in the audience right now, maybe you've done the reachable workspace. If you haven't done the reachable workspace, you sit in a chair. It's a pretty basic chair. And you reach your arm above your head in these different quadrants. And so down the y-axis, we have all these quadrants and these total scores. So what was so fascinating in this data exploration is that which muscles should be involved in reachable workspace. And I think if I was in class, I would raise my hand and I would say, if I could raise my hand, I would say shoulder, stabilizer, shoulder, shoulder, shoulder would be my 100% answer. And it turns out that yes, the shoulder is very involved. So the shoulder is in this blue box over here, but you also have knee extensors and ankle dorsiflexors very involved. And so what's remarkable about this is that, and in thinking about it, it makes a lot of sense, which is if I can point my toes, when I hold something above my head, I kind of like grip like cat claws, like my toes kind of grip the ground and I kind of lean into the front of my feet. And so there's a lot of stabilization that's occurring that is not in the shoulder. And so I use this as an example. So the other piece to notice here is there's an unweighted and a weighted condition. And so it does look like you also have different contribution of either the legs or the shoulder girdle, depending on if you're holding a weight or you're not holding a weight. And so on the one hand, this complexity is sort of horrifying because we now can say that your toes are involved in reachable workspace. I also think that this is helpful because now we can make a model to predict how the change may occur over time. And so I think my next slide is my amazing picture. Anyone wants to hire me to do child's birthday parties or greeting cards, I'm available. Um, so this is sort of what I would say is our current state of how we think about these tasks, which is everyone has a bunch of muscles that are very different, as shown in my people with the little black marks. But right now we look at and we just collapse those down into a pot. And then we look at the tug. We look at their tug performance, and that becomes our outcome pairing. Really, I would like to see a future state where if the first patient has only muscles affected in the dorsiflexors, we measure dorsiflexion. If it's more like the muscles that could be involved in pick up a penny, we use pick up a penny and reachable workspace and so on, so that we're sort of personalizing the muscle, the patient, and the task, because that will be the most likely thing that will sort of pair the stuff up. And let me see, so it's 10:43, so I'm going to buzz through my summary so we have enough time for questions. And reading slides are always difficult because I think we're all like, we tend to read, but I'll just kind of give you a quick overview. So I think what we've learned in the last 15 years is it's just an incredibly heterogeneous disease, and that is the rule, not the exception. So this can vary across subjects, across muscles, across side, and within muscles. And really across the lifespan of the disease, we see all muscles affected. So I used to believe the tibialis posterior was never affected. Well, that is definitely not true. We now, I think, recognize that fat pattern is a very helpful biomarker in starting to try to do prediction. That something about it is useful in knowing how things are going to creep along. I do think strategies should focus on all individual muscles if possible. And this is Sylvia Blemker's statement, no muscle left behind. Like I'm definitely, I'm a convert to the no muscle left behind strategy. And then I think there is sort of a concern that lumping muscles, you have to be a little careful when you start to lump things together because certainly collapsing their identity just adds error. And that is not going to be what you want in trying to have a short trial or a sensitive trial. To go back briefly, this idea of subregion modeling is going to keep boosting sensitivity. And so just to show you, right now, we take two muscles and we say this has 6% at baseline, and now it has 6.25% at follow-up. Tiny. That would be noise. But if you can divide this muscle up into pieces and you look at the piece, now you see that it goes from 15 to 40%. So subdividing muscles is a really great idea because it allows you to look at the area that's changing, not at the rest. And to some degree, this is kind of what I think the Fulcrum trial tried to do with MFI, but this is actually looking at the fat change portion as opposed to looking at the muscle, the limited muscle that's left. I believe that MRI changes will always occur before tasks changes. And so even for something like dorsiflexion, it's going to be likely that you'll see fat change before you'll see a functional change. And as you move towards more kind of bulk tasks like extension, you have to combine muscles into these functional groups. And although that's necessary, it does come at a penalty of not being able to see change as effectively. And certainly with the example of reachable workspace, complex tasks are going to be a much bigger challenge for modeling than dorsiflexion. And so I do think that, as we saw with reachable workspace, these models can help to clarify what muscles are involved. And that may be useful because it may not be always intuitive that the gripping of your toes is going to be a component of raising your arm above your head. And then I wanted to also say, like, the timescales can absolutely differ. And I think we may want to really own that as a field, is that subregion fat is going to change fast. Fat volume is going to change more slowly. Dorsiflexion, strength could change. We kind of have some ideas of that. If you want to then conjoin these two things, like fat and strength, that's going to take much longer. If you want to see a fat change and a complex function change, that's going to take even a lot longer. So depending on how we're designing our trials, could we get away with just looking at fat? I think that's going to be a consideration as we go forward. And then the last things, we're really excited to keep expanding this finite element modeling work to try and understand, like, where is fat? How does it affect function? I think it'll be very, very useful. We're also working on a paper right now. I see that I've got the word generating power estimates. That's what we want. We want the double power estimates for how you can use these things in trials. Like, I think, these are complicated concepts and these are models, but they can generate data that we can really say, if you use an individual strategy, you need 50 subjects at 6 months. If you use a lumped overall body strategy, you need 1,000 subjects at 5 years. So these are things that we can start to really come up with quantitative estimates for. And then lastly, I'll say we're really excited. We're working on the MOVE PED study with kids. We're continually adding data into this data lake. And so really being able to start adding terms like growth in, so we can really understand some other relationships that right now we haven't started to work on. And with that, I'd like to thank, this is a team sport, my many collaborators, and for any of the patients and families that are on the call, thank you so much for lying in the scanner and allowing us to study you. And I'll stop there and just take any questions.
All right. Well, thank you, Seth. You took us from soup to nuts on MRI. And I want to go back to soup a little bit. I'd like to go back to one of your basic premises that was important all the way through your talk, just to be sure that folks understand that premise. And that's about the concept that the pattern of fat in a muscle really matters and affects the progression, perhaps the rate of progression, perhaps it predicts progression, and that's essential. So that's a really key message to come out of this. And we had one specific question about you very clearly said not all muscles are the same. And
Mhm.
I think you showed us the pattern in one muscle, and you anticipate you're going to find different patterns in different muscles. And so one of the questions that came to us, if you look across muscles, are there any correlations between disease progression within a particular muscle or a muscle group, and the origin, their fetal origin or the fetal development, or the amount of use that they get?
Mhm. Yeah, I think it's a great question. And I think that, one of the things I think that we've really benefited as a field from starting to look at this stuff from a biomechanical standpoint is that, I'll make 2 points. One is that lean muscle is super important. lean muscle volume and lean muscle function. And these are, I think, important concepts because as a field, we've really been focused on fat fraction as the biomarker of interest. And, this is something that if we're just talking to someone that studies muscle force, like fat has no force generation potential. It's only lean muscle volume and the patterns of things that are there. So I think there is a lot of likely things to learn about, is the area of maximal force generation in the muscle most vulnerable to progression. I think that is going to be a very useful area of inquiry. And I don't know enough about, muscle cell lineage, those sorts of things to comment. I think it's a very good question.
Yeah.
I do think that one of the things that we've been talking a lot about is issues of like compensation and hypertrophy and atrophy and sarcopenia as you get older. And, if you have foot drop on the right side, you're going to hike your hip when you walk because you need to raise your foot. And so those muscles are going to hypertrophy, but they're not necessarily, it's a compensatory response to just normally what muscle does. And so I do think that it's an interesting problem to kind of chase some of these things around. We do need probably better strength data as a field. Like I think really, really focused strength data may help us to answer some of these. There's some, I didn't show the slide, but from the FSHD these batteries where we do a lot of MMT, those are actually quite well correlated to lean muscle volume in assemblies, like, the extensors in the leg. But you can't, that's obviously a lot of muscles that deal with knee extension. And so you got to put them all together to get that measure. So I think one of the challenges is that because muscles work in these groups, it can be a little difficult to disentangle some of these questions.
Right. And so I'll stop you there and I'll just say, so I think your answer to the question is we don't have enough information.
Thank you, Lucienne.
You're welcome. He described a lot of fat infiltration or fat fractions. He's also saying that lean muscle volume. So how much muscle is actually remaining how much muscle material is remaining in each individual muscle is very important to its function, perhaps the most important. And so we still have lots of work to go to understand if, use is going to drive progression, if origin of muscle is going to drive progression. And there's a lot of work yet to be done. But what he did show us, and I'll just reiterate that point, is that fat patterns appear to be able to help us to predict progression. And I'm going to take him to a later stage question here, which I think is also important, and I'd like people to understand, which is that we believe, Seth for sure believes, I believe, I think the community believes that MRI is going to be valuable for selecting patients who might predict during the time course of a clinical trial. And using MRI, therefore, could make us more successful in running our clinical trials because we'll have identified muscles or muscle groups that are likely to progress. And then we can measure that progression when they're getting no drug versus when they are getting a drug. And so, Seth, I'll ask you to reiterate that point that you made and kind of exemplify that a bit more.
Yeah, I mean, I think that, I would like to envision a future where, if you're a patient and you enter a trial, let's say your name is Billy, we find out which muscles are likely to, we scan your body, we get your muscle profile, and we make a prediction based on you of how you're going to change over the trial. And then essentially deviations from that prediction using the random forest or something like it is the drug effect. And I think that too often we're sort of hoping that there'll just be some, universal tonic of the drug, which will affect dorsiflexion, and that will be enough with our sort of double blind, our two group design. And I think that heterogeneity is enough of a problem that is going to limit what trials can see. And so I think that, I would like to believe that we can start to use this personalized data to create an at-risk set for Billy, an at-risk set for Jenny, and then we're following those MRI features over time. And we're also able to follow tasks that should change based on those, sort of that MRI phenotype. One other comment, I would love, and we're certainly hoping to do it in some of the data that we have, but we'd also love to start taking some of these blood biomarkers that have been developed and seen by Avidity and others and correlate those with the burden that we see on MRI, because at least at this point, that's the best tell we have that things are changing. And so I'm very hopeful that we can actually see like how those things map together.
Okay, can I, we have two more questions. I just want to see if we can, one individual who may need to have a hip replacement, and they're asking, will the amount of fat that are in the muscles that are involved in the recovery affect their rate of recovery or affect their ability to recover? Do we know enough about presence of fat and how it impacts muscle recovery, either from surgery or from other types of damage?
I would think the only, not knowing how much of a total hip I'm glad I don't totally know what connects and disconnects, but I do think that, if you imagine an intact muscle system where you have all muscles are healthy and they screw together whatever they're going to screw together, I presume that you have sort of a normal course of recovery from that. And I think if you have fat in your hip that affects abduction, for example, you may have to do more rehabilitation because you're starting at a sort of a more compromised point. Um,
And I think that's a reasonable and good answer. And then the last few minutes,
thank you.
people are one. Thank you, Seth. Sorry, Seth and I know each other very well, so we've worked together for many years.
Fine. Harassment is fine. I like it. Yeah.
Okay. So how close are we to having MRI biomarker testing for standard of care and monitoring, not in a clinical trial, but just in clinical visits?
I think it's a great question. I mean, I think we look to some of our colleagues in Europe and Australia, it's just more routine. I think that there are disease models like chronic osteomyelitis where MRI is used to follow the disease. And I think it's a great goal. I think we should reach for it. I think that it definitely, we have work to do in terms of being able to tell you quickly, like, oh, this is what the pattern means and how it's going to potentially affect your fall risk or your function. But it's a great, I think it's a great call out. And I think it would be very helpful both for patients and for doctors and for scientists to just know what's going on.
Yeah.
So
Having the whole body MRI in the clinical trials has demonstrated to us very clearly how valuable that data is to understanding disease. The global FSHD group from Australia has started to work to make the data from a whole body MRI available to patients, and that's something that folks in the US are now requesting. And the society is hoping we can work with Springbok to facilitate that kind of information. And I believe we are out of time. I hate to say it because I think this is a really complex area and there's a lot of lessons here. And I think that we could do a bit more work after this to draw out some of those big question and big answer areas and try to make them a little more clear for like lay people who are really trying to understand how MRI really explains and exemplifies the disease. So, Ashley, I think we could do a bit more work here, and that will be our goal for the future.
Absolutely. Thank you, Dr. Friedman. Thank you, Dr. Ronco. This has been absolutely wonderful. We really appreciate you taking the time to explain all of these concepts and answering our community's questions. But also thank you to our community for being here and for really being an active part of our community and showing that you want to understand this so that you can continue to help us with research as it's advancing, really be research ready and eventually treatment ready. So thank you. Before we close, I did just want to give you all a reminder that our next FSHD University will take place next week on Tuesday, September 23rd at 1 p.m. And it will focus on preventing falls, moving with balance, awareness, and intention. It'll be presented by Frank Hanley. And we also have three FSHD universities in the month of October that I recommend people sign up for so that they can attend and hear about smart home technologies for individuals with FSHD, the FSHD Global Innovation Hub, and then navigating Medicare, maximizing your benefits with FSHD. So a lot of exciting things coming up. Thank you to all. We hope that you will join us soon at one of our many events, and we'll see you at our next FSHDU. Take care, and thank you again, Dr. Friedman.
Yeah, thank you all. Appreciate it.
Thank you.
Bye-bye.
Bye.
Bye.